Here is a question that does get asked often enough in a drug development. When a biodegradable polymer breaks down inside the patient's body, where do the breakdown products actually go? With PLGA, the most widely used biodegradable polymer in injectable drug delivery, the answer is that lactide and glycolide monomers accumulate at the injection site, get absorbed systematically and metabolized through known biochemical pathways. That part is documented and understood. What's less well managed is the residual monomer load that the polymer carries before it ever reaches the patient. The unreacted lactide and glycolide from the synthesis that your standard characterization might not be measuring. Hello, I am Raghuvar Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, and we work with pharma and biotech teams every day to build the analytical and regulatory foundation for injectable drug products. Today, in this session of our parental biomaterial series, we are going to unpack the residual monomer challenge in PLGA, what it is, why it matters for the patient safety, and what a regulatory ready testing programs looks like. PLGA, the polymer inside the modern injectables. PLGA has been commercially successful for a reason. It's FDA approved, biodegradable, biocompatible and tunable. You can dial the degradation rate from weeks to years by adjusting the lactide to glycolic acid ratio and molecular weight. Products like Lupron Dapot, Risperdal Consta, Vivitrol and Biduren have proven that PLGA microspheres can deliver the drugs reliably over 1 to 6 months from a single injection. That's a remarkable clinical achievement, but this success has sometimes outpaced the analytical framework that supports it. 30 plus marketed products, and the field is still debating what the appropriate residual lactide acceptance criteria should be. That gap is where regulatory risk lives. What PLGA is and why chemistry matters. PLGA is synthesized through ring opening polymerization of two cyclic dimers, lactide and glycolide. The word cyclic dimer matters. Lactide is a dimer of lactic acid, glycolide is a dimer of glycolic acid. And when polymerization is incomplete, which it always is to some degree, those unreacted dimers remain in the final polymer as the residual monomers. The LA-GA ratio you choose shapes everything downstream. A 50-50 PLGA degrades in 1 or 2 months, fast enough that monomer accumulation at the injection site becomes a real local tissue concern. An 85-15 formulation for a 6-month implant gives you more time, but a longer window for the residual monomers to migrate into surrounding tissue. The chemistry isn't just a formulation choice, it's a safety profile that needs to be characterized. So where residual monomers come from? So where exactly are residual monomers coming from? The primary source is incomplete polymerization. Some fraction of lactide and glycolide feed never incorporates into the polymer chain. A second source, one that surprises teams unfamiliar with polymer processing, is depolymerization during manufacturing, heat, shear stress during milling or spray drying, and exposure to moisture can break polymer chains back down into monomeric species. Third pathway is the synthesis solvent itself. Ethyl acetate and dichloromethane are common PLGA synthesis solvents, and they can form solvent monomer complexes that survive downstream purification steps. And then there is post-processing degradation. The moment your PLGA formulation contacts an aqueous environment during fill finish or reconstitution, hydrolysis begins. By the time the product reaches the patient, the residual monomer profile may already have changed from what you measured at the batch release. 3 safety risks you cannot assume away. Why does the residual monomer load matter from a safety perspective? So 3 distinct mechanisms. First, local tissue toxicity. When lactide and glycolide accumulate at the injection site and begin to hydrolyze, the local pH can drop significantly, low enough to cause tissue irritation, delayed wound healing, and in insensitive tissues, sterile abscess formation. This is not theoretical, it has been observed in preclinical models and reported in post-marketing surveillance for depot microsphere products. Second, immunogenicity. At elevated local concentrations, degradation products can act as molecular adjuvants, priming an unintended immune response. This is a particular concern for biologics co-delivered in PLGA system. Third, and most immediately actionable for your development program, regulatory rejection. The FDA and EMA treat residual monomer characterization as the required CMC element. If it's missing from your submission, it's a deficiency, and that deficiency will delay your program. What regulators expect you to measure? Regulators have been increasingly explicit about what a complete PLGA characterization package looks like. Residual lactide and glycolide monomers sit at the top of the list, quantified with appropriate sensitivity, measured against justified acceptance criteria, and tracked across the stability time points. Residual solvents are mandatory under ICH Q3C. Ethyl acetate is a class 3 solvent with a 5000 ppm limit, but dichloromethane is a class 2 solvent with a much tighter 600 ppm limit, and your GC method needs to resolve both. Molecular weight distribution, MWMN polydispersity provides process consistency evidence and correlates with degradation rate predictions, and free acid content, the lactic acid, glycolic acid generated by hydrolysis in the formulation matrix, rounds out the picture. The pyramid here is not about which tests are optional, it's about where to invest the most method development effort first. Where PLGA CMC packages have stumbled. The cases are instructive. Generic PLGA microsphere ANDAs have received complete response letters specifically citing inadequate characterization of residual lactide relative to the reference listed drug. The argument is straightforward. If your PLGA has a different residual monomer profile, you haven't demonstrated pharmaceutical equivalence. Novel PLGA PEG nanoparticle INDs have triggered type B meeting requests because unlike established microsphere systems, there are no predefined regulatory benchmarks. The FDA expects the sponsor to derive and justify their own safety thresholds, and in the post-approval space, several depot products have received post-marketing commitments requiring long-term monitoring of inflammatory markers at the injection site. At Resolve Mass, we have supported sponsors through each of these scenarios, helping build the analytical and justification packages that unblock submissions and resolve CRL deficiencies. The pattern across all of them is the same. Inadequate residual monomer data always costs more to fix after submission than to build correctly during the development. The 5-step PLGA residual monomer testing workflow. What does a rigorous testing workflow look like in practice? Step 1 is polymer characterization, understanding the LAGA ratio, molecular weight, and expected degradation profile before you design a single method. Step 2 is method selection, and this is where teams most often make the wrong call. Lactide and glycolide are the best quantified by GCFID or GCMS. They are cyclic, semi-volatile species. Free lactic acid and glycolic acid, being polar and non-volatile, require HPLC UV and LCMS. These are different analytes requiring different methods. Step 3 is method validation to ICHQ2R2, specificity, linearity, LOQ, accuracy, precision, and stability. Step 4 is batch testing across multiple manufactured batches and at stability time points. And at step 5, the one most development teams underinvest is in limit justification. The FDA expects you to derive an acceptable residual monomer limit from a safety perspective, not simply report a process capability number. So what should you take from this discussion? PLGA degrades by design. Residual monomers are an inherent product attribute, not a manufacturing imperfection, and they need to be characterized as such. Lactide and glycolide require GC methods. Standard HPLC will not adequately quantify them. Regulators now treat residual monomer limits as mandatory CMC content and not supportive data. And for generic PLGA programs, comparability to reference products' residual monomer profile is a core element of demonstrating pharmaceutical equivalence. In next coming videos, we'll go deeper into residual solvents in PLGA synthesis, specifically why ethyl acetate and dichloromethane need more than a standard USB method, and how your headspace GC conditions can make or break the sensitivity you need. If you are developing a PLGA microsphere, implant, or nanoparticle program and need a CRO partner who understands both polymer chemistry and the regulatory expectations around it, Resolve Mass Laboratory supports injectable drug product programs from early polymer characterization through to NDA and ANDA submissions. Thank you so much for watching this session until now. Goodbye for now and see you in the next one. Until then, goodbye.